Assalamualaikum and good morning. My name is Nur Fitria binti Ahmad Munir and my matrix number is 23DBT 20F1036. And the other group members are Fatin Nasuha binti Abdul Rahman and Tanuja Kumaran. So we are from group 7D and the title of our research is The Awareness of Polytechnic Nilai Students on the Consumption of Fermented Food in the Diet such as tempeh. So, for the first one is the introduction. Fermentation is one of the oldest food processing technologies known to man, as well as one of the most cost-effective methods of food production and preservation. Okay, fermentation. What is mean by fermentation? Fermentation is an anaerobic process in which microorganisms like yeast and bacteria break down food components into other products. This gives fermented foods their unique and desirable taste, aroma, texture, and appearance. Through this fermentation process, fermented food has the potential to carry additional health benefits, especially when they contain probiotics and prebiotics. But in this research, we emphasize tempeh as a fermented food. Tempeh. Tempeh, what is mean by tempeh? Tempeh is a traditional Indonesian fermented soy food made from soaked soybean and cooked using rhizopus mold in salt-free using aerobic fermentation. On the island of Java in Indonesia, tempeh is considered a stable source of protein that has been consumed for centuries. Okay, Rhizopus oligosporus. What is the function of Rhizopus oligosporus in the production of tempeh? Okay, Rhizopus oligosporus is the fungus used to ferment tempeh. The dense cotton mycelium formed during fermentation binds the soybeans together to form a dense cake. Tempeh is becoming popular, and today more and more grocery stores have begun to carry tempeh products. This is because it is known to reduce cholesterol, increase bone density, decrease menopausal symptoms, and promote muscle recovery. In addition to these amazing benefits, tempeh is easy to prepare, delicious, high in protein, and rich in manganese, copper, and phosphorus. So, I move to the next, which is the problem statement. This research reviews a problem faced by the unhealthy lifestyle we lead, including food intake. According to the latest estimates from the World Health Organization, which is WHO, Malaysia has the highest obesity problem in Southeast Asia and the sixth in Asia. In the last 45 years, fat and sugar intake has increased by 80% and 33% of respectively. Then, by changing our lifestyle in food intake can improve health performance. From our research, some Polytechnic Nilai students are unaware that eating plant-based protein is one of the healthiest ways to consume it. Unfortunately, many vegetarians suffer from the protein deficiency due to the lack of protein boosting options and awareness. Tempeh is a good choice for plant-based proteins. Tempeh also can be used as a nutritious meat replacement. And the fermentation process has already helped convert some of the protein into amino acid, reducing the amount of work required by the digestive system. So next, proceed with the research objectives. There are three research objectives in this research. So first, to give awareness to Polytechnic Nilai students on how important healthy food is such as tempeh. Number second, to study the benefits of tempeh as functional foods. And lastly is to differentiate the benefits of tempeh using different alternative substrates like soybeans and mung beans.
Hi everyone, my name is Tanuja Kumar. My metric number is 23EBD20F1047. Today I would like to continue our presentation on literature review. So first is tempeh. Tempeh is a fermented food made of main soybeans and is a nutritional, affordable and sustainable functional source of proteins. Globally, tempeh is widely accepted fermented product, although there is a growing body of literature on tempeh. Most research has focused on unfermented soybeans, thus the impact of tempeh fermentation on the biological properties of soybeans has been legally left centered. This review concluded that sufficient evidence exists in the literature supporting tempeh fermentation as a low-cost, health-promoting, and sustainable food processing technology to produce protein-rich foods using various beans, legumes, and grains. This comprehensive review suggests further study are needed on tempeh fermentation and its impact on human health. Research and standardization of non-soya tempeh, assessment of food safety improving modification in tempeh production system, and initiative supporting the sourcing of local ingredients in tempeh production. Next is effect of R. oligoporosis. R. oligoporosis was examined for its effect on the structural and functional features of fermented soybean. After 24 hours of fermentation, the amount of total free amino acid produced in soybean significantly increased to 1.20 gram per 100 gram of soybean. This was done to boost the flour's use potential. Starch extracts were produced from flour derived from fermented soybean, employing R. oligoporosis throughout a 72 hours fermentation period. Standard procedures were used to determine the psychological properties of the starch extract, as well as the amylolysis, amylopeptine, and pasting qualities. Most of the psychochemical properties studied examined increased tense with increasing fermentation length with the expectation of the reconstruction index, which shows declining tendency while the peak breakdown and final viscosizations all dropped. The swelling power and solubility increase slightly as the fermentation period increases. So next is fibrinolytic enzyme. Fibrinolytic enzymes are one of potassium enzymes that can be used to break down fibrin in cardiovascular therapy. Fibrinolytic enzyme can be sourced from microorganisms. For instance, Bacillus comes from traditional fermented food. Bacillus bacteria has a function as a proteolytic procedure. Tempeh is one of traditional Indonesian's traditional fermented food made from soybean, which is fermented by Rhizopus species mushroom, which also produce fibrinolytic enzyme. The purpose of this study is to determine whether the enzyme produced from fermented Vigna eleusis tempeh by Rhizopus or rhizolysis have fibrinolytic activity and to determine the effect of temperature, pH, metal ions and inhibitors of the activity of the fibrinolytic enzyme fermented by Vigna eleusis by Rhizopus. This method used in this study was quantitative. The method design of this study was purely experimental. There was a replication randomization and control or composition group. The result of the study revealed that the activity test for fibrinolytic enzyme using positive tyrosine stance contaminated the enzyme protease. Meanwhile, the characterization analysis, it was found that the crude extract of red bean tempeh could reach the optimal condition at 50 degrees 50 Celsius and pH value of 5, while in the metal activator, magnesium plus 2-methyl showed increased activity of fibrinolytic enzyme, and PMFF inhibited shows strong resistance, indicating the serine protease group. That the crude extract of red bean tempeh has potential fibrinolytic enzyme activity and can be used as an alternative to therapeutic therapy. So next is the benefit of mung beans. Mung beans fermentation by lactic acid bacteria is an intriguing effort to generate innovative functional food. Mung bean milk was produced fortified with sugar and fermented with Lactobacillus platyrum B1-6 in this investigation. Fermented MBM was studied for its microbial communities, protein properties, peptide profile, and also ACE inhibitors efficiency. Sucrose concentration, inoculum concentration, incubation temperature, and duration were found to influence the final lab population using a one factor at a time technique. The maximum lab population of 8.96 log CFU per ml was obtained under optimum circumference using response surface methodology experimental design. Most of mung bean protein were destroyed during the fermentation according to erythropoiesis profile, with hydrolysis percentage ranging from 49 to 69 percentage. Reverse phase high performance liquid chemotherapy revealed that larger or more hydrophobic peptides vanished while smaller or more hydrophilic peptides increased. At the end of the fermentation, L-plantarum B16 fermented MBB had a considerable higher ACE inhibitors activity, around 67.5%. Overall, our work showed that MBM may be usefully lab carrier, and that fermented MBM by L-platyrum B16 had a high potential to be used as functional new food. Next is the benefit of soya bean. Eating protein-rich products instead of animal can lower serum concentration of total cholesterol, low-density lipoprotein, and also triglycerides. So next is the benefit of isoflurone. Isoflurone has been proposed to be the active component responsible for the beneficial effect of soybean foods and appear to work in conjunction with the protein to protect against cancer, cardiovascular disease, and osteoporosis. Most of the research activities on the benefit of soybean foods have focused on the role of these isoflurans play in diseases prevention or treatment. However, there is also some evidence that the benefits are attributable to certain peptides or protein fractions from soybeans. This review will be focused on some of the potential mechanisms whereby soybeans extract their protective effects against heart disease, cancer, and osteoporosis. So next part will be the hypothesis. Next is hypothesis. First hypothesis is based on the studies, PNS students are unaware about benefit of tempeh consumption. Second point is tempeh consumption will reduce calorie intake, avoid malnutrition and obesity. The third hypothesis is there is no significant difference between soya bean and mung bean tempeh. Both of the tempeh provide high protein and nutrients.
Assalamualaikum, my name is Fatina binti Abdurrahman. So today I will continue with methodology. So our method is survey and secondary data. Survey and questionnaire are commonly used in quantitative social research to get the information that we aid in understanding the need of individuals on specific topic. Surveys are also used to get quantitative data on items in a population. Survey also was conducted on Polytechnic Nilai students. So, using this method, we can achieve our objective such as to provide awareness on the importance of healthy food intake such as tempeh. So, next point is secondary data. So, by use the information that has been collected by other researchers, we can get the information such as a benefit of tempeh and fermented food history. So for the expected result is students who eat tempeh will be healthier than students who do not eat tempeh. For the another one is students who do not eat tempeh will be healthier than students who eat tempeh. So I will continue with budget. So budget that we do in while doing the research. So the activity is questionnaire development. So the cost is 0 ringgit, and instrument distribution. The cost also 0 ringgit. Statistic software and also final test. So the total cost is 0 ringgit because the survey is entirely conducted online. So there is no funding set up for it.
So this is the references in our research.
This is the Gantt chart. So the activity we are conduct is a literature review, proposal, sample preparation, analysis, thesis writing, final presentation, correction, final submission. So this is the Gantt chart, and we can have a look on that. We are start our project on from May until December.